Today we will discuss about a posterior pituitary hormone, that is arginine vasopressin, or simply the vasopressin or antidiuretic hormone. Another posterior pituitary hormone is the oxytocin. It is a small molecule, it's an oligopeptide having 9 amino acid sequence. The gene for the ADH synthesis, it is located at the chromosome number 20. Chromosome number 20. Okay. It is synthesized by the magnocellular neurons of supraoptic nuclei and also the paraventricular nuclei of the hypothalamus, the major source being the supraoptic nuclei. It is synthesized at the cell body of the magnocellular neurons, and those are transported to the nerve terminals which are located at the posterior pituitary for the storage. The storage of the vesicles at the terminal end of the nerve, causing the swelling, and which is known as the herring body. Right? So the storage of the vesicles causes expansion of the nerve ending, and this is called the herring body. Now, like any of the peptide hormone, it is synthesized as a pre-pro hormone, which is also known as the pre-pro presophysin. Now, this pre-pro presophysin, it will be converted to the pro presophysin in the Golgi body, where it will be stored into the vesicles and will be transported along the exoplasmic flow to the nerve terminal for the storage. Ultimately, in the vesicles, it will be broken down into equimolar concentration of antidiuretic hormone and the neurophysin 2. The neurophysin 2 function, it is to transport the ADH to the nerve terminal and prevents its leakage. Now coming to the regulation of vasopressin or ADH secretion. The main regulator of the vasopressin secretion, it is the plasma osmolality. Now plasma osmolality, it is the number of dissolved particle in a fluid. It is the number of dissolved particle in the fluid. So indirectly you can say it is the concentration of the solution, and it is expressed as the total number of solute particles per kg, usually milliosmol per kg. The normal plasma osmolality, actually it averages between 280 to 285 milliosmol per kg. Now the hypothalamus has the specific region over it, the anterior hypothalamus. Now to be more specific, it is over the OVLT. What is the full form of OVLT? It is the organum vasculosum of lamina terminalis, and also over the SFO. SFO, it is the subphronical organ. Now whenever there is increase in the plasma osmolality, suppose the OVLT, it will fires and stimulate the supraoptic and the paraventricular nuclei, causing increase in the ADH secretion. The second most important regulator of ADH secretion, it is the blood volume and the blood pressure. Now whenever there is decreased in blood volume or decreased in blood pressure, there will be increase in the ADH secretion. The receptors which are located at the low pressure side of the circulation, particularly over the atrium and the large pulmonary vessels, they detect the fullness of the circulatory system. So basically they act as the volume receptors. So whenever there is decrease in the blood volume, they fires. Whereas the receptors which are located at the high pressure side, like those are located over the aortic arch and the carotid sinus, they are the baroreceptors and the stress sensitive, and they essentially detects the changes in the pressure. So whenever there is decreased in the mean arterial pressure, these baroreceptors will fire. Now if you draw the structure, like it is the central nervous system. Here I am drawing the brainstem. This is the cerebellum. Now I will be drawing the receptors which are located over the heart, and those are located over the carotid sinus. That means these are the baroreceptors. Now from which the information both from the low pressure and the high pressure, the sensation it will be carried, or the afferent it will be carried via the glossopharyngeal and the vagus nerve at the medulla, up to the medulla, at the cardio-regulatory center, that is the nucleus tractus solitarius. Now from here the nerve, it will supply the paraventricular and the supraoptic nuclei in such a way that whenever there is decrease in the blood volume or there is fall in mean arterial pressure, there will be increased stimulation of the supraoptic and the paraventricular nuclei. Another regulator of the vasopressin secretion is the angiotensin II. Whenever there is fall in BP, there will be stimulation of the renin-angiotensin aldosterone system, ultimately producing increased amount of angiotensin II. Now there are specific areas over the hypothalamus which has angiotensin II receptor. Specifically, those are located over the OVLT and the SFO. Now the SFO, it is the full form subfollicle organ, and OVLT, it is the organum vasculosum of lamina terminalis. Now whenever there is AT2, it will bind to the specific receptor situated over the OVLT or SFO, that causes ultimately increase in the ADH secretion, as well as there will be increase in the thirst. There will be thirst stimulation. Now this OVLT and SFO, along with two other structure, that is area postrema and the posterior pituitary. So posterior pituitary, area postrema, OVLT and SFO, they forms the circumventricular organs. These organs are remaining outside of the blood-brain barrier. So any change in the systemic vessels, it will also affect these structures as they are located outside the blood-brain barrier. Other regulators that increases the ADH secretion are the nausea, vomiting, pain, standing position, hypoglycemia, increase in the temperature, and stress. The factors that decrease the ADH secretions are ethanol, decrease in temperature, atrial natriuretic peptide, thyroxine, and alpha adrenergic agent. ADH or vasopressin has two types of receptor, the V1 and V2. V2 receptors are located at the kidney and it is related to the function of water reabsorption. V1 receptor is again subclassed into two groups, V1A and V1B. V1A, it is located at the vascular smooth muscle, and activation of it causes the vascular smooth muscle contraction, whereas V1B, it is located at the hypothalamus and it is related to the secretion of the corticotropin releasing hormone. Now if you look at the mechanism of V1, it is a receptor, it is a 7-pass receptor, G-protein coupled receptor, and inside it is linked with the GQ protein. GQ, it activates the phospholipid C, which breaks down the membrane associated phosphatidyl inositol biphosphate into two components. The first one is the inositol triphosphate and the another one, it is the diacylglycerol. Now inside the vascular smooth muscle, it has a rich internal store of calcium, which are stored inside the sarcoplasmic reticulum or endoplasmic reticulum. This sarcoplasmic reticulum has specific receptor for the IP3. When the IP3 binds with that receptor, calcium ion will be released from the sarcoplasmic reticulum into the cytosol. This calcium ion, now it binds with the calmodulin to form calcium calmodulin complex. This calcium calmodulin complex, it now activates the myosin light chain kinase. This myosin light chain kinase, it phosphorylates the myosin light chain and activate it. Activated myosin light chain, it interact with the actin molecule and causes the muscle contraction, vascular smooth muscle contraction. The another component of the PIP2, that is the diacylglycerol, it activates the protein kinase C. This protein kinase C, it activates and phosphorylates different enzymes and the protein, which is responsible for the CRH secretion function of ADH. The V2 receptors is present over the renal tubule at the distal convoluted tubule and the collecting duct. This portion of the renal tubule, it is selectively permeable to water depending upon the presence of antidiuretic hormone or the vasopressin. So whenever the vasopressin, it comes out of the circulation and it acts over the V2 receptor, which is a 7-pass receptor and G-protein coupled receptor. It binds with the receptor and inside it, it activates the G-stimulatory protein. This G-stimulatory protein, in turn, it will activate the adenyl cyclase enzyme, which converts the ATP molecule into cyclic AMP molecule. This cyclic AMP molecule, it activates the protein kinase A. This protein kinase A, specifically, it phosphorylates the water channels aquaporin 2, which are packed inside the vesicles. So these water channels get phosphorylated. These water channels are specifically of aquaporin 2 molecule. Now this phosphorylated aquaporin 2 channels packs inside the vesicle, it will now move towards the apical membrane of the cell along with the help of the molecular motor, which is depicted in green color along the microtubule, this orange colored thing. So the dynein molecule helps the vesicle to move along with the microtubule towards the apical membrane. Now when the vesicle reaches the apical membrane, it will now get fused with the apical membrane, so that these aquaporin 2 channels are fused with the apical membrane. Apical membrane, it is the side of the cell which line towards the renal tubular lumen. So the water is flowing in this direction, and if the aquaporin 2 channels are inserted inside this apical membrane, water, it will now go inside the cell. At the basolateral side of the cell, there is already aquaporin 3 or aquaporin 4, and the presence of these water channels is not dependent upon the presence of antidiuretic hormone. So the water now, it will pass through and through of the cell into the extracellular space and ultimately into the circulation. Now what is happening in long term, if there is chronic excessive intake of water, what is happening in psychogenic polydipsia, there is already excessive fluid inside our body, and the body doesn't need the excess water more, right? So there will be pKa mediated decreased synthesis of the aquaporin 2 and the aquaporin 3 molecule, so that the diluted urine, it will pass through the renal tubule, and there is no more reabsorption of the water from CD and DCT. So what are the different functions of the ADH? Now from the mechanism of action, we have already discussed that via the V2 receptors, which are located over the distal convoluted tubule and the collecting duct, ADH help in water reabsorption, and which restores the extracellular fluid volume, right? So if there is decrease in the extracellular fluid volume, it tries to restore that ECF volume. Now via the V1 receptor, particularly the V1a receptor, it causes the vascular smooth muscle contraction, that is the vasoconstriction. But you have to keep in mind that this vasoconstriction action of the vasopressin, it happens during the supraphysiological concentration of the ADH. So it is happening during the supraphysiological concentration of ADH. Whereas the water reabsorption function of the ADH, it is happening in the physiological concentration of ADH. Now via the P1b receptors, we have already discussed that helps, that those are present at the hypothalamus, and it causes increase in the release of corticotropin releasing hormone, ultimately increasing the ACTH concentration. ADH, it may act as a neurotransmitter in the brain, where it facilitates the memory. It may also act at the area postrema, and via the action over the area postrema, it decreases the cardiac output. It also helps in the glycogenolysis in liver. In different parts of the central nervous system, this antidiuretic hormone, it may act as a neurotransmitter. Two drugs have been developed against the ADH receptor. The first one is the conivaptan, which is specifically a V1a and V2 receptor antagonist. The another drug is the tolvaptan. Tolvaptan, it is specifically the V2 receptor antagonist. So if we block the V2 receptor, there will be excessive excretion of water without the sodium ions, as the V2 receptor function, it is only to reabsorption of the water, not the sodium. So there will be, if there is excessive water loss but without sodium ion, there will be increase in the sodium ion level, and this may be helpful in the treatment of hypervolumic hyponatremia. This tolvaptan, it is the US FDA approved drug for the treatment of syndrome of inappropriate ADH secretion.